Precision for Women est un podcast dédié à la médecine de précision et à la santé des femmes. Vous y retrouverez régulièrement des témoignages et des interviews des femmes et des hommes qui constituent l'actualité de ces deux disciplines. Je suis Norbert Nabé, votre hôte. Soyez les bienvenus. Aujourd'hui, nous avons le plaisir d'accueillir le professeur Martin GOETTE, qui est chercheur en biochimie médicale et chef du département de recherche de gynécologie obstétrique de l'université de Munster en Allemagne, pour un entretien qui aura donc lieu en anglais. Bonjour Martin.
My name is Martin GOETTE. I'm a professor of medical biochemistry and a basic researcher, and I'm head of the research laboratory of the Department of Gynecology and Obstetric at the University of Munster, Germany. And I've been doing research on endometriosis since about 20 years. I am coordinating a European project on endometriosis called Trendo. I'm also coordinating a large German project on endometriosis and fertility funded by the German Research Ministry. My research interests are very basic, so we want to understand the molecular mechanisms of the disease, and in recent years we have focused on two topics. It's the role of endometrial stem cells in endometriosis, and more recently functional analysis of microRNAs in the disease.
All right. These two mechanisms are two in the middle of many others, and there's a big question mark on the physiopathology of this disease. And maybe after a short overhaul of the main hypotheses, can you develop the two fields in which you are involved?
Yes, the major hypothesis how endometriosis is developing date back to the last century, and a very influential hypothesis is the concept of retrograde menstruation, by which menstrual blood and endometrial tissue is distributed via the fallopian tubes and reaches ectopic locations in the ovary and in the peritoneum. There are additional theories like coelomic metaplasia or embryonic rest, and these describe more developmental biology-related mechanisms. So the coelomic metaplasia states that there may be a transformation of peritoneal cells into endometrium-like cells due to exposure to, for example, chronic inflammatory stimuli. While the embryonic rest theory states that cells from the embryo may have remained in the female body, and they can also give rise to endometrial tissue. And in addition to these theories, it's thought that endometriotic tissue may be distributed via blood vessels or lymphatic vessels. And there are also some theories like the tissue injury and repair theory, which states that damage due to a hyperperistalsis of the uterus may lead to local damage and to increased estrogen production. And these classical theories can explain how endometrial tissue ends up at the wrong location, at the so-called ectopic location. But these theories have in modern times been complemented by more molecular and cell biological concepts. And one of these ideas is that stem cells could be an underlying cause for endometriosis, because stem cells can basically live forever. They have unlimited proliferation capacity, and stem cells also have a high developmental plasticity, meaning that they can develop into other types of tissues. And the stem cell hypothesis is compatible with all of the classical theories of endometriosis. The stem cells could be distributed via the menstrual blood in the retrograde menstruation. The stem cells could also be involved in the coelomic metaplasia or the embryonic rest. So this is a very attractive idea that these cells could be the origin of some endometriotic lesions. And from the cancer field, we know that stem cells are also much more resistant to therapy because they express multi-drug resistant proteins, which can basically eliminate drugs posed to the cells. And they also have a more efficient DNA repair. So they are basically more resistant, and therefore it's very difficult to treat.
Is there a link between these two physiopathological or biological process, cancer and endometriosis?
I would say that there are many similarities, but I have to state endometriosis is a benign disease, so endometriosis is not cancer, but it shows because there are not like these typical mutations that you find in tumors which are driving tumor progression. So this is not present in endometriosis, at least not to this extent. However, there are many secondary aspects which are very similar in cancer and in endometriosis. One aspect is that endometriosis can grow in an invasive way, meaning it can penetrate into tissues. It can also metastasize in a way, it can reach different locations. There is also angiogenesis involved, like the growth of blood vessels towards the endometriotic lesion and the blood supply of the lesion. This is present both in endometriosis and cancer. And of course, the unlimited proliferation may be mediated by stem cells, but also by other factors such as estrogen, for example, which promotes cell proliferation. So I think on a secondary level, there are many parallels between malignant disease and endometriosis.
And maybe on this specific site of the cancer, can we imagine one day that a mechanism like immunotherapy could have an effect on this disease?
I think in the endometriosis field, we could really benefit from the developments in the cancer field. For example, if you have a molecular target, like let's say a cell surface receptor, which plays a role in cancer, and you have a drug against this target, you could use this knowledge to treat endometriosis patients, for example, because then the drug has already undergone clinical trials for the cancer application, and it's much easier to get this drug approved for endometriosis. However, this also has some limitations. Cancer is, of course, a life-threatening disease, and some treatments are very harsh, but you tolerate them in a cancer setting because it's a question of living or not. However, for a benign disease, you cannot be as radical as you are with a life-threatening disease.
Yeah, sure. Well, the second way of your research are microRNA. So what's up with microRNA and endometriosis?
Yes, so several studies have shown that microRNAs are dysregulated in endometriosis. So microRNAs are small non-coding RNAs, and they are a physiological mechanism by which a cell can negatively regulate mRNAs. And as mRNAs are responsible for all kinds of physiological function, the regulatory power of microRNAs is very strong. And my laboratory is mainly focusing on molecular mechanisms, and we try to find out how certain molecules work within a cell. And we have done quite a bit of work on microRNAs in cell culture models. So what we could show for several microRNAs is that they are inhibiting invasive growth of endometriotic cells in the laboratory. We could also show that they are regulating signaling pathways important for inflammation, which is also a factor in endometriosis. It's a chronic inflammatory disease, and we could show that some of these microRNAs can block this inflammatory phenotype. And what we also showed is that some microRNAs can block a process called epithelial to mesenchymal transition. And this is a process which makes cells more invasive. And actually this mechanism is also a key mechanism for the invasion of cancer cells.
Sure.
So again, there are parallels at the cellular level between endometriosis and malignant disease.
Your exploration, your research are based on the tissues.
We are mainly working with cell lines and cell models. So we partially use patient biopsies to culture stroma cells, so a part of the endometriotic lesion. And then we treat these cells with microRNA precursors and look for the effect that this microRNA has in a given cell. And there are also some cell models which were originally derived from a patient biopsy, but which have been genetically manipulated, so you can culture them in the laboratory basically forever. So it's a more standardized model that we are using there.
You do that on chips?
No, we don't do it on chips yet. So previously we used standard cell culture on tissue culture plates and tissue culture flasks. But now we are taking the next step to move into three-dimensional models. And we are now combining epithelial cells and stroma cells in defined extracellular matrices, and we're trying to recapitulate the organization that we find in tissues. We are also working with organoids derived from patient cells, And we even have a bioprinter which allows us to artificially print endometrial tissues. And this is used to study the effect of microRNAs, for example.
Is it different from different kind of phenotype, classic localization of the disease, or do you work on a universal model of the development of the disease?
We have worked more on a universal model, or mainly on the superficial endometriosis, but I now received funding from the German Research Ministry, and we now want to expand our study to look at the differences between the different locations.
Maybe we can make a small link with the cancer, because you told me that you are working also on different types of cancer, especially ovarian cancer, because we found in certain kind of ovarian cancer endometriosic lesion. So
yes.
Do you make the link between these two diseases?
Yes, well, in a parallel line of research, we have been focusing on breast cancer for a long time, so there is no real connection to endometriosis. But now we have a certain shift in interest, so we are also now working on endometrial cancer and ovarian cancer. And epidemiological data show that there is a link between ovarian cancer and endometriosis. But this work is just starting. But what we are doing is, if we are evaluating the effect of a microRNA, we usually do this in parallel. We study the same microRNA in endometriosis and in the respective cancer. And what is very interesting is that sometimes we see differences in the effect that the microRNA has, because one microRNA has many targets within a cell, and the effect of a microRNA at the functional level will depend on which mRNAs are expressed in a given cell type. For example, if an ovarian cancer cell does not express a target of a certain microRNA, but the endometriotic cell expresses this target, then the microRNA will only affect one particular pathway in one disease, but not in the other.
Well, maybe we can talk about the link, this famous link between fundamental research and practice.
I'm a basic researcher, so I try to understand how a disease is developing and which molecules are responsible for a certain disease. But the understanding of these molecular mechanisms also is important knowledge to develop drugs. So our basic research work is also defining potential new drug targets. But in general, it's said that a discovery, an exciting discovery in a basic research laboratory takes about 10 years until a drug is on the market. This includes, of course, all the preclinical assessment in animal models, cell culture models, and the different stages of clinical trials, which are, of course, required to ensure that a drug is safe. Therefore, this long time is not surprising. However, I think with the microRNAs, we may have some very exciting new drug targets, or the microRNAs themselves may be interesting drugs when they are chemically modified. And again, we can learn from the cancer field and other diseases, because pharmaceutical companies are already developing inhibitors for certain microRNAs for other diseases. And once such drugs are approved, they could also potentially be used for endometriosis if we find a mechanistic association with endometriosis.
What these molecules could target, which mechanism, whether it's inflammation, whether it's pain, whether it's...
Yeah, theoretically, they could target all of these processes because the action of microRNAs is very fundamental. So microRNAs are regulating mRNAs, and mRNAs are the information for building proteins. So basically, every conceivable process in a cell, or nearly every conceivable process in a cell, could be theoretically regulated by microRNA. And usually the microRNA is not only targeting one molecule, but several molecules. So the effect of a microRNA-based drug can be predicted to be quite strong. On the other hand, there's also an increased danger of having side effects if you target many molecules at the same time.
Sure.
But this can be evaluated in preclinical studies to ensure that such a drug is really safe.
Well, you talk about the future with the therapeutic target, other field or collaboration, or
Yeah, well, as I said, I'm already actively collaborating with many people in the endometriosis community. For example, I'm coordinating a European project called Translational Research on Endometriosis, Trendo, which has 12 partners. And we have, for example...
It's a European project?
Yes, so we have partners from Estonia, for example, from Sweden, from Germany, even from Argentina, and each group has its own specialty. For example, the Argentinian group is focusing on animal models, but they also are very strong in natural therapies of endometriosis. The Swedish group is strong in finding links between cancer and endometriosis. The Estonian group is a biotechnical company. They are very good at transcriptome analysis. And my laboratory in Germany is very strong at the mechanistic evaluation. And we also have experts from Maastricht in the Netherlands who are working on hormone metabolism. So this Europe-wide project gives a strong collaboration and allows us to study endometriosis at multiple levels. It's already the second project on endometriosis that I'm coordinating. So we had another predecessor, which was called MOMENDO, Molecular Mechanisms of Endometriosis. And the first one was more basic research oriented. And now the second project, which is currently running, is already going more into application. So with the additional collaboration with partners in Poland and Slovenia, we want to combine the forces of all members to also look for diagnostic biomarkers in a multi-omics approach, like looking at mRNA levels, at hormone levels, and at protein levels. As we are many partners in different European countries, we have access to a much larger patient collective than an uni-national project could have.
Sure. I wish you a very large success.
Yes,
for
thank
this,
you.
Because it's very important, and thank you. Talk is very clear and it's fascinating. Thank you very much, Martin.
Yes, thank you for having me.